Enzyme? What is that?
What is enzyme? Enzymes are proteins that assist the conversion of substrates to products without being altered themselves. Enzyme activities are mainly influenced by temperature, substrate concentration, and pH. Then, enzyme also highly specific, which it can only bind to substrates that fit well the active site. Enzyme, also known as biological catalyst, which accelerates reactions by lowering the activation energy of reactants. The figure below shows that catalyzed reaction has lower energy barrier compared to uncatalyzed reaction. So catalyzed reaction can occur at a faster rate compared to uncatalyzed reactions at the same condition. Now let's look at the enzyme structure. Enzymes may be simple or conjugated proteins where it has 4 levels of structure, which is primary structure, secondary structure, tertiary structure, and quaternary structure. But mostly enzymes are found in quaternary structure. Then enzymes are also consist of cofactor, catalytic site, and coenzyme. Holoenzyme is referred to a fully active enzyme that is composed of apoenzyme and cofactor. Active site is a region that binds to substrate, cofactors, and prosthetic groups. It contains residue that helps to hold the substrate. It also consists of 2 types of sites, which is binding site and catalytic site. The role of binding site is to choose substrate and binds it to the active site, while catalytic site will perform the catalytic action of enzyme. Then, for cofactor is a non-protein molecule that exists in 2 types, which is organic and non-organic. Organic cofactors can be further divided into prosthetic group and coenzyme. Prosthetic group is a metal ions such as magnesium and ferrum that is tightly bound to enzyme, while coenzyme is a substance that loosely bound to enzymes such as vitamin B12 and folic acid. Now let's move on to enzyme classification. Enzyme can be classified into 6 types, which are based on the type of reaction that they catalyze. The first one is oxidoreductase. Oxidoreductase is an enzyme that catalyzes the transfer of electrons in form of hydride ions and hydrogen atoms. The second one is transferase. Transferase is an enzyme that catalyzes the transfer of functional groups from one molecule to another. The third one is hydrolase, which is an enzyme that catalyzes the cleavage of bonds by hydrolysis. The fourth one is lyase. Lyase is an enzyme that catalyzes the dissociation of molecules by removing groups or addition of groups to double bonds. The fifth one is isomerase, which catalyzes the transfer of groups within a molecule to yield isomeric forms. The last one is ligase. Ligase catalyzes the combination of 2 molecules by condensation coupled to ATP cleavage.
Now, how does enzyme work? Basically, enzyme will bind with substrate to form enzyme-substrate complex. Subsequently, the previous substrate will form new products. Meanwhile, the enzyme will be reused in the next cycle. There are 2 models used to describe the way enzymes interact with substrates, which is the lock and key models and also the induced feed model. First one is the lock and key model. In this model, the active site has a rigid shape, and only substrate with the matching shape can fit. Hence, the substrate is the key that fits the lock of the active site, and this explains the enzyme specificity and the loss of activity when enzymes denature. The second one would be the induced feed model, where the initial interaction between the enzyme and the substrate is weak, and this rapidly induces changes in the shape of the active site to fit the substrate like a mold. Moving on to the enzyme specificity, where it is about the selectivity of enzyme to their substrate. In total, there are 6 enzyme specificity, and the first one would be the bond specificity. The enzymes are specific to substrates having similar bonds and structure. For example, the alpha amylase hydrolyzes alpha-1,4 glycosidic linkage. The second would be the substrate specificity, where the enzymes are specific only to one substrate and one reaction. For example, lactase only hydrolyzes beta-1,4-glycosidic bond. Moving on to the third enzyme specificity, which is the group specificity. The enzymes are specific to a bond and groups surrounding the bond. For example, pepsin hydrolyzes the peptide bond. Next, we have the optical specificity. The enzymes are specific to substrates and its optical configuration. For example, the L-amino acid oxidase acts only on L-amino acids. Then, we have the geometrical specificity. The enzyme can act on different substrates having similar molecular geometry. For example, alcohol dehydrogenase can oxidize both ethanol and methanol. Lastly, we have the cofactor specificity, where only correct combination of substrate and cofactor allows enzymatic reaction. For example, lactase only hydrolyzes beta-1,4-glycosidic bond. Let's look into the enzyme utilization in the food industry. There are three main groups of food enzymes, which are the carbohydrates, proteases, and lipases. Carbohydrates catalyzes the breakdown of carbohydrates into simple sugar. The subtypes for these carbohydrates include amylase, cellulose, lactase, and pectinase. Proteases basically break down proteins into amino acids. For example, bromelain, cysteine, and papain are widely used in the food industry for various applications such as brewing, tenderization of meat, and also the coagulation of milk. For the lipases, it breaks down fat into fatty acids and glycerol. It is mainly used for flavor development in dairy products and processing of other foods containing fat. In addition, lipases is also used as flavor development agents in butter and margarine and to prolong the shelf life of various baking products. Now, let's look further into the enzymatic browning in food. Enzymatic browning is the oxidation reaction that takes place in some foods, mostly fruit and vegetables, causing the food to turn brown. The polyphenol oxidase, which can be found in the fruits, changes the phenols into melanin, which has a brown color when it is exposed to oxygen. However, this enzymatic browning, which is unfavorable, can be stopped using various methods. Let's look into the physical methods to prevent enzymatic browning. Firstly, the thermal treatment. Heat will inhibit the enzymatic activity as the enzymes, which are composed of proteins, are denatured. Using low temperature can also prevent enzymatic browning, as the lowering temperature by pre-cooling, refrigeration, or freezing will suppress the enzymatic browning. Aside from that, enzymatic browning can also be prevented by substituting the air with an inert gas such as the nitrogen or CO2. One of the chemical methods to prevent enzymatic browning is by the acidification or reduction using antioxidants. Acidification or reduction using antioxidant will inactivate the PPO by lowering the pH. Aside from that, we can also use the copper chelating agents, which suppress the PPO activity by binding to metal cofactors in the PPO enzyme structure.
Peroxidase is an enzyme found in a wide variety of organisms, from plants to humans to bacteria. Active enzymes in fruits or vegetables can cause post-harvest deterioration in quality and nutritional value. This deterioration occurs when the products are frozen. Fruits and vegetables are usually blanched before freezing or canning. The experiment effect of blanching on peroxidase enzyme is done. This is because blanching conditions need to target at most heat-resistant enzymes. Peroxidase is one of the most heat-stable enzymes in plants. It is a good indicator for blanching adequacy, since heat treatments sufficient to inactivate peroxidase also will inactivate most of other enzymes. In this experiment, potato is used as the sample. Firstly, the potato is cut into the form of wedges. Then it is submerged into 300 ml of boiling water at 3 different beakers with each beaker being boiled at different duration. The potato is then rinsed with cool running water, which indicates completion of blanching process. A piece of sample weighing about 5 grams is cut into small pieces and then transferred into a mortar containing a small quantity of sand and distilled water for grinding process. The mixing process is done by adding guaiacol solution and hydrogen peroxide. Next is the results of the experiment. There is significant color changes that can be observed. The one with the shortest time of blanching turns into brown, while the sample that has been blanched for 5 minutes shows slight color changes. We can relate this with this equation. Hydrogen peroxide is broken down into water and oxygen by the peroxidase enzyme. Then the oxygen reacts with guaiacol that will produce brown color. Hence, this explains why the sample that is blanched for 1 minute is dark brown in color. The enzyme actively breaks down the hydrogen peroxide, producing more oxygen to react with guaiacol. Further analysis can be done using spectroscopy. This video is taken from Rungnarun Sasanatayart that tests the peroxidase activity using broccoli as a sample. After doing the same procedure, they did further analysis using spectrophotometer to calculate the absorbance. Spectroscopy is the study of absorption and emission of light and other radiation by matter as related to the dependence of these processes on the wavelength of the radiation. Next is the schematic diagram of the spectrophotometer. Firstly, it can be seen that tungsten lamp and the D2 lamp acts as the light source and then reflected to the monochromator through the slit. The emitted light passed through another slit and reflected to the sample and the reference sample. The intensity is then detected by the photodetector. Next is the working principle. Each monochromatic beam is split into 2 equal intensity beams. The intensities of these light beams are then measured by electronic detectors and being compared. The intensity of the reference beams, which should have suffered little or no light absorption, is defined as I0. The intensity of the sample beam is defined as I. UV visible spectrophotometer is commonly used for analysis of enzymatic activities. In this type of spectrophotometer, the wavelength of UV extends from 100 to 400 nanometer and the visible spectrum from 400 to 700 nanometer. The advantages of using this method that it is low in cost, fast and simple.